Hi everyone, good morning.
Hi.
Hope you all are doing well. So today we will start with molecular epidemiology and typing. Yeah, today's lecture will be covering that. And what we are going to learn today is basically we'll look at phenotypic and genotypic identification, some molecular methods, epidemiology, methods of typing, and genomic epidemiology as well, little bit, uh, very briefly. So, we all know about bacterial identification and subtyping, and we all know like there are various ways of identifying um, all sorts of pathogenic bacteria or microbes. So, the number one way is obviously using microscopy, which is you're doing your gram staining and other procedures. You can also have like biochemical tests. Other than that, there can be immunological tests that can be performed. Phenotyping using phages, for example, can also be performed. And lastly, there are molecular approaches. Obviously, there are lots of molecular approaches. So I've highlighted some methods like DNA fingerprinting, PCR techniques, which is RAPD, REP-PCR, AFLP. Um, and then you have also got other methods like pulse field gel electrophoresis, PFG, MLST, and whole genome sequencing. So for pathogen identification, as we know, this is a very classic step in microbiology. And you can see that in the chart that is there on your right hand side, where you have the culture, you can do gram staining, you're observing that as gram positives, gram negatives, um, rods, cocci, cocobacilli, and performing different biochemical tests. And based on the tests, you are grouping them into certain categories or in certain clusters. Um, in biochemical identification, obviously, you can do a biochemical test in order to find out. A simple example of a biochemical test is sugar fermentation. You can have sugar fermentation that happens. But other than that, you can also have indicator media. You can see in the picture over there on the bottom right, where you can see different types of colonies or different colored colonies that are present. So, so uh, that is nothing but chromogenic agar, right? Because chromos is color, genes is giving rise to. So something it is giving rise to color. So that is the chromogenic agar. Or it can also be, you can also have selective media. You have selective media, you have got differential media. Selective meaning they select for certain type of organism. Differential because they can differentiate between different types, lactose fermenter, non-fermenter. Enriched because they have a lot of nutrients in it. That's why they are enriched. So these names are there. Don't be very worried about what these names are. It is whatever the name is there, that is the meaning of that medium. So we all know like microscopy and all is very cheap, it's easy. Everyone does gram staining. Gram staining was invented in the 1800s. It is one of this classic methods that is still used even in UK. I'm pretty sure in India as well. It is still used. It is never going to go out of date. Um, So some negatives can be it is subjective, sometimes not enough distinction to identify species because obviously if you have infection from Staph aureus or Staph epidermidis, During gram staining, you will just see cocaine clusters because it is staphylococci, right? You are not able to differentiate at the species level whether it is aureus or whether it is epidermidis because the species are very distinct. Excuse me. So, as I said earlier, microscopy example of that is gram stain. You can also have fluorescent microscopy is also there sometimes, but this is the most common one. And everyone knows what is a gram staining, right? You have the briefly to tell you what is the procedure in that for those who don't know. You have got the gram positive cells and you have got the gram negative cells, right? So these two cells are there, two different types of cells are there. The positive cells, they stain purple or violet in color, and gram negative cells that are there, they stain pink in color, right? But principle behind this